A warm welcome to all. Today, I would like to introduce our speakers, Dr. Banappa S. Angar, who is a e-scientist, Pharmacology and Toxicology Division from Indian Institute of Traditional Medicine. He holds MPharm PhD degree in pharmacology. He has guided 18 MPharm projects and he has also guided 14 students in MD Ayurveda, 2 from MTech Bioinformatics, 5 students in MPH, 4 short-term research trainees. He has 22 research publications and also 1 patent to his credit. He has handled 6 research projects from RGHS, ICMR, DHR, CCRAS, etc. He has participated and attended a number of national and international seminars and workshops and undergone numerous trainings programs in DST, ICMR, a few of them. He is life member, South Asian College of Clinical Pharmacology, and affiliate of American College of Clinical Pharmacology, founder member, Translation Biomedical Research Society, India. He's member Karnataka Ayush Society, National Ayush Mission, Government of India. He's also a member of Society Enthopharmacology. A warm welcome to you, sir. Next, we have Dr. Chandrasekhar. He is a professor and HOD in pharmacology from HSK College of Pharmacy, Bagalkot. He has completed master's and doctoral studies PhD in pharmacology. Dr. Chandrasekhar is an active researcher who is constantly working in the field of research with 14 years of experience, who is constantly working in the field of research with 14 years of experience and has nearly 19 years of experience in teaching. He has published more than 75 research papers and peer reviewed in national and international journals. He has also procured many research grants from DST, VSGT, RGH, etc. as a principal investigator. Dr. Chandrasekhar has visited USA, Chile, South America, and Thailand for presenting his research papers in the prestigious national and international conferences. Moreover, he has been an invited as a speaker and a chaired session at the national and international conferences. He is a member of Board of Studies post-graduation in RGUHS, Bangalore. He has guided many students for their PhD post-graduation projects under RGUHS. His areas of expertise include screening of new chemicals, entities against neurodegenerative diseases, and anti-inflammatory. His MPharm project is sponsored under project program from KSCST, Indian Institute of Sciences, IICS, Bangalore. And this project work is awarded as the best among the Life Sciences Project of the Year 1997 to 2000. He has also received 2 travel grant awards under Young Scientist from Department of Biotechnology, Government of India, New Delhi. I welcome you, sir. Now, the instructions for the participants are as follows. While your presentation is being presented, you need to mute yourself. And during the question answer sessions, you need to switch on your videos and audios. Thank you. We have first presenter, Ms. Lakshmi, with track ID T1PCOL001.
Good morning, everyone. Myself, Lakshmi Patanjali from Department of Pharmacology, Haley College of Pharmacy, a constituent unit of Haley Academy of Higher Education and Research, Belgavi. Poster entitled is Cynodon dactylon L attenuates beta 142 amyloid content in scopolamine-induced amyloid rats. And the poster number is T1TCOL001. Thank you so much for giving me an opportunity to present the poster. As all of us know, dementia expresses the features of neurodegeneration that leads to memory impairment and behavioral changes, which interferes with the work and person's ability to perform the routine activities. The pathological hallmarks for Alzheimer's disease, which is a part of dementia, are hypothesized, such as interruption in the cholinergic neuronal system, amyloid pathway, or hyperphosphorylation of protein tau and its accumulation in the brain. As per the Unani system of medicine, Cynodon dactylon was reported as a heart and brain tonic. However, scientifically, so far there is a paucity regarding the research of Cynodon dactylon effects for its potential to treat dementia. Hence, this study evaluated the Cynodon dactylon hydroquinolic extract on beta 142 amyloid content of scopolamine induced cognitive impaired rats. The methodology includes that experimental rats of both the sex or either sex are used within the body weight of 200 to 300 grams, and 6 animals were used in each group. Institutional Animal Ethical Committee approval was taken, and rats were acclimatized to before experiment initiated. Scopolamine hydrobromide 1 mg per kg through IV route was used to experimental rats except normal control for 30 days. To the another group, donepezil 3 mg per kg through oral route was used as a standard control, and to the rest group, Cynodon dactylon extract at the range of 100, 200, and 400 mg per kg through oral route were administered as a treatment for 15 days, except scopolamine and normal control groups. Later, the rats were sacrificed to isolate the brain for the measurement of rat amyloid levels according to the manual of Steinfitch enzyme immunosorbent assay procured from bioassay technology laboratory science. As we can see, in the Steinfitch ELISA method, the plate will be pre-coated with beta 142 antibody, and in the sample, if the beta amyloid 142 is present, then it binds to the pre-coated antibody. Later, the bioattenuated antibody was attached to it, and then the added streptaviridin will bind the bioattenuated amyloid level. The unbound streptaviridin HRV is washed in the washing step. Later, the substrate solution will try to produce the colors if the sample is having the beta amyloid level. Then the stop solution was added to measure the content at 450 nanometers. And the results are analyzed using the mean and standard error mean. Statistically, Tuckey's post hoc multiple comparison test was used, followed by ANOVA to check or analyze the statistical difference of various groups. And they've considered significant if the p-value is less than 0.05 or less than 0.01 or 0.001. As we can see in the results, scopolamine administered rats showed a significant increased levels of beta amyloid concentration in picograms per ml as compared to normal control, whereas groups treated with the donepezil and the Cynodon dactylon extract at different levels showed a significant decrease in the beta amyloid content as compared to scopolamine group. This may confirm the traditional claim of Cynodon dactylon in modern therapeutic targets for dementia. Further, cognitive studies may explore the clinical relevance of the use of the Cynodon dactylon, including its in vivo studies
Miss Lakshmi, are you there?
Hello, ma'am. Yeah, yes, ma'am. I'm there.
Please switch on your video
Yeah.
and we can proceed with the question answers.
Yeah, thank you.
Lakshmi,
Hello, sir.
audible?
Yes, sir. I'm audible. Good afternoon, sir.
Good afternoon, Lakshmi. It's a very good presentation.
Thank you, sir.
The work actually, it is of a high importance in
Yes.
the increasing the disease prevalence in the coming days also.
Yes, sir.
So here, apart from that, what are the other parameters which you have measured?
Yeah, sir. Actually, I have measured the parameters in the in vivo studies, including the cognitive impaired rats in the elevated plus maze and as well as the Morris water maze, including the acetylcholine level through the ex vivo model.
Okay. So, are there any corroborating the evidences of showing this the dose dependent effect which is shown in this?
Yes, sir. In the acetylcholinesterase enzyme estimation, possibly I could find that there is a decrease in the concentration of the acetylcholinesterase level as compared to the scopolamine induced rats. And meanwhile, in the in vivo studies where the experiments were conducted regarding the Morris water maze and the elevated plus maze, there was a quite improvement in the long term and as well as short term memory. So
What
this
is
was
the
the report.
what the plant, whether it is easily available, and what is
Yeah, yes,
what's
it's
the translation?
easily available, sir.
Okay. So the findings. What is the translational value of your findings?
So basically, earlier days in the traditionally in Unani system of medicine and as well as in the Ayurvedic system of medicine, this was used by many of the doctors availably. But nowadays, if we just compare it with the modern therapeutics, we can just take the lead molecules and then translate them into a bedside, or we can translate them into a preclinical studies and then lead with a lead compound available, and then we can make it in modern therapeutic usage in treatment of dementia and some other CNS disorders. And especially, I could find that this is also important in antipsychotic and as well as in the Parkinson's disease treatments.
So yours is a whole extract the study, no? I think the fractionation is not done and characterization is also not done.
Not yet done, sir. So it's just based on the
That,
extract study.
yeah, that will add on a value, in fact, to this
Yes,
study.
sir. Yeah. That will be giving a further studies, sir.
All right. Okay. All the best.
Okay. Thank you, sir.
Over to Sivangi.
Chandrasekhar sir, are you there?
Hello,
Yes, sir.
is it audible?
Yes, sir, you're audible. Please proceed.
Okay. Hello, I have
Yes,
some
sir.
basic questions.
Yes, sir.
Okay. What is the rationale behind selection of this plant?
Basically, if we just go back to the earlier days, like Ayurveda or Unani system or Siddha system of medicine, these have been having a research related to this, but there were paucity or there is a lack of knowledge regarding this in the preclinical study. So that's why I carry forwarded this work, sir.
Okay, then in previous studies, which part of the plant they have used?
They have used the stem, they have used the root parts. I have used the whole parts of the plant.
Whole plant you are used.
Yes, sir.
Then you are subjected for extraction with which type of solvent?
I have used the maceration process, which included the 70s to 30 ratio of hydroethanolic extract, sir.
Okay, what is the rationale behind that, and which possible chemical constituents you are going to expect it in this extract?
Yeah, so basically it's a mixture of the flavonoids, steroids, the polyphenols, tannins, and some of the carbohydrates were also included. And in the maceration process, whatever the hydrophobic agents that or you can say the water soluble phytoconstituents were solubilized in the water, and those who are solubilized in the ethanol part, they were extracted from that.
Which activities are probably has the more
Basically,
important basic pharmacological activities what you are observed, no?
yes, sir.
Which chemical actions are probably involved in this type of activity?
Yeah, basically the flavonoids and the steroids, especially, they were having a lot of role in giving the action.
Steroids, you are told. Which kind of steroids? Do you have any some ideas? Name
Phytosteroids.
some
The phytosteroids. So basically, still have to go with the fractionation process and as well as isolation. Then we can come to know.
You have done this activity, which is the possible mechanism. Not you are not done, but which is a possible mechanism.
A
It is going to be known.
possible mechanism might be either through the cholinesterase enzyme inhibition and including with the beta amyloid, probably by decreasing the content of that. And as well as including the neuroinflammation, it can reduce the neuroinflammation.
The scope of study is very good. Hope you have to be carry out these all activities.
Yes,
So
sir.
finally, you have to prove the mechanism of actions and all.
Okay, sir. Thank you
And,
so much.
good luck. Thank you.
Thank you.
Next, we have Sai Pratima with track ID 002.
A very good afternoon to one and all. I'm Rachel Sai Pratima of Krupacon College of Pharmacy. Today I'm going to discuss about my research topic, assessment of anti-mutagenic activity of Malus domestica by micronucleus assay in Swiss albinomites. I'm under the guidance of Ms. Kesana Bhavani, Assistant Professor at Krupacon College of Pharmacy, Bangalore. Coming to introduction, first what is mean by genotoxicity? These are the chemical agents that damage the genetic information within a cell, causing mutations which leads to cancer. These events play a major role in many malignancies. Thus, identifying genotoxic or mutagenic effects is an important factor for the risk assessment of a substance. Here, the plant I have selected is the Malus domestica, which is a pomaceous fruit that is apple, which belongs to Rosaceae family. It is a perennial plant, and it is most widely grown in India. Coming to collection, authentication, and extraction of the plant material, the fruits of Malus domestica were collected from the local market and authenticated. The fresh fruits were taken and peeled off to the flesh or the pulp. 1 ml of distilled water was added and blended at high speed. The blended homogenate was strained, centrifuged, filtered, and the extract was subjected to the phytochemical screening to identify the chemical constituents. Next, the phytochemical constituents that were identified in the fruit are carbohydrates, proteins, and amino acids, alkaloids, tannins, phenols, steroids, terpenoids, glycosides, phenols, fixed oils, resins, and coumarins, respectively. And the method I have done in this research is the micronucleus assay. Here, 8 to 10 weeks old Swiss albinomites were used of weight 25 to 30 grams, and the animals were approved by the animal committee. The animals were grouped into 5 groups of 6 animals each, and the first group serves as a control, which contained 6 animals and were given distilled water and saline solution. And the second group were treated with the inducer mitomycin C for 2 consecutive days. And for the third and fourth groups, the animals were treated with Malus domestica extract and the inducer mitomycin C of dose 50 mg and 100 mg respectively. And the fifth group of animals were given standard melatonin along with the mitomycin C inducer. After 48 hours of the treatment, animals were sacrificed, femur bones were collected, the proximal ends of the bones were opened, and the bone marrow was flushed into a centrifuge tube filled with phosphate buffer saline. The bone marrow suspension was centrifuged at 3000 rpm per 10 minutes. The supernatant was discarded and pellets were resuspended with PBS. A drop of cell suspension was kept on a slide and made smear. Then the smear was dried, stained with Zimsa stain, and the cover slip is placed with DPX. Nearly 1000 polychromatic cells were identified or observed at a 1000x RP, at 1000x magnification with oil immersion. And the micronuclei that are observed in the control test and standard drugs were given in the form of pictures. Results in the present study, the mutations were induced by mitomycin C. The parameters evaluated are the total NC count, total PC count, MNPC count. Due to the presence of resins polyphenols, the extract shows significant reduction in the mutations that are induced by the mitomycin C. The extract will show the reduction in mutations by reducing the ratio of NC to PC count. The potency of the extract was compared with the standard drug melatonin. Through the results obtained, the extract of Malus domestica are significantly proved that having the antimetagenic activity by using the micronucleus assay. Thank you for giving me this opportunity. Thank you, everyone.
Ms. Pratima, please be ready for question answers.
Yes, ma'am.
Please switch on your video.
Pratima, am I audible?
Yes, sir. Good morning. Good afternoon, sir.
Good afternoon. It's again a very good study which I have come across. I appreciate that effort is in this direction, but I would like to know that what is the rationale behind selecting this particular the fruit for your study?
Because this apple, that is Malus domestica, has many antioxidants and phenols, and its phytochemical constituents are very good. This antimetagenic or anti-cancer, it has many anti-cancer properties. So I have selected this plant.
The dose which is shown this efficacy, is it I mean translatable to this the regular practice?
Yes, sir.
What is the amount which a person should take this fruit? The quantity of the fruit which should be consumed to get protection from this genotoxicity?
Nearly 100 grams to 200 grams.
So that is the translational value. Always should think in that angle. The study is no doubt the wonderful concept. It is the routinely used or easily available the medicinal plants or the nutraceuticals, if they are protective against such toxic effects of these chemicals, which day to day everybody is getting exposed to these toxic agents. The findings has a relevance to that human health, in fact, but we should think in that angle, that how much quantity. So what is the extraction method which you are used? Which method you have used for the extraction?
I just extracted the fruit pulp and I just boiled in ethanol, sir.
Okay, boiled in ethanol.
Yes, sir.
Okay. So okay. Congratulations for your presentation. Thank you.
Thank you, sir.
Hello.
Yes, sir. Good afternoon, sir.
Is it audible?
Yes, sir.
So there's a question saying I have also doubt. So I have selected the extraction procedure. What is the rationale in behind selection of extraction procedure?
Because generally it's a fruit, the storage, because the storage conditions, like it won't be stored like above 2 months. so that it is easily available food, so that I can prepare if I need more extract.
Then how you have extracted? You told boiling in ethanol. How?
I have extracted the fruit pulp,
Okay.
and I have added ethanol, and then I
Okay.
boiled till it comes to thick slurry.
Okay. How much temperature you have kept for boiling?
Sorry, sir, I have forgotten.
No, that's the question you told. Directly boiling, how it's possible with ethanol.
Water bath.
Okay. Then what are the possible active constituents which are soluble in ethanol? Do you have any literature you have done for this, or you have done any phytochemical investigations?
No, sir.
Okay. You tell
Actually,
me which
I have referred the previous article, and I have selected this plant.
Okay. Now, according to you, which active constants is responsible for detoxification? You told they are going to
The
reduce
flavonoids,
the
phenolic compounds such as flavonoids and anthocyans. These are responsible for this activity. And on antioxidants also, sir, like quercetin and all.
No, every plants have an antioxidant, but your level of his activity is different. So their simple mechanism of antioxidants, it is only the main responsible. Is there is any other possible mechanism? Okay, no, leave it, not a problem. Just you go through that, okay? Just go through it.
Yes, sir.
And thorough review of literature, and you can go for further scope of activity is good. So you can do the best work, okay? Good luck.
Okay, sir. Thank you.
Next participant is Mr. Lingaraj with track ID 003.
Hello, hi everyone. Myself is Lingaraj from the Department of Pharmacology, University Kumareshwar College of Pharmacy, Bagalkot. Today I am going to presenting my research work on the neuroprotective activity of Matricaria icodata against the Alzheimer's disease. As we know that many of the plants are going to get used for various diseases such as the neural disease like Alzheimer's disease, Parkinson's disease, or some of the anti-inflammatory diseases or the analysis disease or analysis activity. Yeah. For that, today I am going to get presenting a study evaluating of the neuroprotective effect of Matricaria reputata against the person-induced Alzheimer's disease in the rat models. First, we go to know that the background of this Alzheimer's disease is that it is one of another progressive, irreversible, insidious neurodegenerative disease, which is similar to that of a coronary artery disease of the heart. And one of the most important of these pathological involved in Alzheimer's disease is that amyloid betas, as well as the tau hypothesis. These are the two main important hypotheses, along with the cholinergic hypothesis, as well as the oxytocin and metronine hypothesis are going to get involved based on the amyloid beta hypothesis because of an amyloid beta subprotein, as well as tau hypothesis based on the special type of protein called as a tau protein. Next, I just go to the plant profiles that is called Matricaria reputata, and it is also going to referred as one of the star among the medicinal species, and it's going to get belongs to a family called Asteraceae, as belong to the genus of the Matricaria, as well as this species belong to called as the reputata. The parts used for this is for this current study is called as the flowers as well as next the methodology that involves the research work is called as Alzheimer's disease is going to get induced by the intracerebroventricular administration of an ecosystem in the lateral ventricles by using of the 25 microliter of the microsyringe in the STDs. Where it is divided into 5 groups. So the first group of 5 sham group is administered with the 5 microliter of an artificial cerebrospinal fluid for in the intracerebroventriculars. Along with the 28 days, they are going to get the distal waters, followed by there is a control group which is going to be called as the colchicine induced group, which is administered with the 5 microliter of the ACSFs that containing up to 50 microgram of the colchicine drug, and this group is treated up to 28 days with the distilled waters, followed by the remaining 3 groups of 100 mg, 200 mg, and 300 mg per kg. They are going to treat it with the 15 microgram of persistence that is dissolved in 500 ml of ACSF, along followed by there is a 28 days treated with the methanol extract of the matrix reported they are going to be treated, followed by the NL's behavior parameters are going to get assessed up to the 28 days at an interval of 7 days, that is 0 days, 7th day, 15 and 28 days. Followed by the 28th days, there is a biochemical estimations are going to get conducted, such as total thiols, catalase, LPO, GSH, as well as SOD and pollination rates. And in this research work, I just done an acute toxicity of this plant up to the dose of 3,200 mg per kg shows that the not toxic effective and there is no any mortality are going to get found, hence dose is safety safest dose. Hence there is 100 mg and 200 mg and 300 mg per kg body weight are going to get selected for the studies. Next I just go for the results. In these results of Moritz water based observation tables on the day 14 and day 28 at a dose of 200 and 300 mg per kg of treatment which shows significant activity compared to the control groups, shows that there is a decrease in the escape latencies in the treatment groups as there is an increase in the escape latency in the control group when they are going to get compared with the sham group, which is going to cause a normal groups. Next, I just go for the mean transfer latency, which is called as a plus measure. In plus measure, there is a control group is going to get increased in the mean transfer latency of that is a plus measure activity compared with the sham group. Where treatment groups are going to results, there is decrease in the time record of the transfer latency is decrease in the treatment groups, that is 200 as well as 300 mg per kg. Along with the 28 days, there is a 100 mg per kg. So it's shown a significant activity. Next, and open field, there is a rest. In a rest, there is a control group shows that there is an increase in the rest period of times in control groups and compared with the treatment groups. There is a significant activity shown in the treatment groups. There is an increase. There is a increase in the control groups. There is a decrease in the treatment groups. Next, go for the biochemical estimation and is total thiols. In total thiols, the treatment groups like 100 mg, 200 mg, and 300 mg shows a significant activity. And compared with the control groups, as a result, there is an increase in the total thiols in the treatment groups. And the lipid peroxidation is going to get decreased in the treatment groups compared to the control groups. As well as in catalase, shows that there is significant increase in the 300 mg per kg in the catalase activity compared to the control groups. Next, in the glutathiones also shows a significant activity with the decrease in the control groups when compared to the sham groups, and treatment shows the increase in the GSH levels compared to the control groups. So finally, there is a pollination estimation shows that the treatment groups are going to get increased at an interval of 2 minutes when they are going to get compared with the control groups. The control groups show the significant decrease when compared to the sham groups. Finally, just go for the discussion and conclusions, which shows that this is one of the very best model for the selection of for inducing of persistent condition Alzheimer's disease, and followed by the treatment with the methanol extraction of matrix reported as show that there is a input conjugation and decrease in LPO and restored the DSH total thiols catalase and increase in the acetylcholine transferase level. And this is about the research work. And I just thankful to the principal and management of HSC College of Pharmacy Bagalkot for supporting me and providing me necessary facilities for the studies. And also I just thankful to the Krupacon College of Pharmacy, Bengaluru, for organizing global or online conference and workshop on drug development 4.0 emerging technologies. Thank you, one and all.
Please be ready for question answers.
Lingraj.
Yes, sir.
So
yes, sir.
It's a busy schedule. Even the slides is very busy. That now you will put a lot of content in that. Was unable to see the minute details of it. Anyway, I would like to know that what is the rationale behind this selecting this plant particularly? So can you?
Sir, actually the plant is the Matica respirata, which was one of the neuroprotective activity in different models, like the global cerebral scheme model as well as in Florida induced and oxidative stress model in our institute. So I just selected this plant. I also go through the chemical constant of this plant, which containing of bioflavonoids as well as the flavonoids, the cumbrians is going to contain, and also the rutinine is going to important for this some of the oxidative stress in this model. So I just selected this the plant for the activity.
Now, are there any traditional use or kind of any hints that it has this protective effect? neuroprotect?
Means the basic of the plant is.
Go ahead.
Sir, actually, it is having it shows that it in one of the literature is mentioned. It shows that of neurotonic.
In which system? Because I never come across such things. Maybe I missed it. So would like to know that, is it because of just the phytochemicals which are found? Phytochemicals are in general, it are no flavonoids and all are there in most of the plants.
Yes, sir.
That may not be the correct guide for your selecting this plant. So are there any the traditional uses track, use of history of use kind of a thing?
Just I did not go through that, sir.
Okay, it's okay.
Okay, sir.
uh, anyway, that's a good study. Congratulations.
Okay, thank you, sir.
Hello.
Sir.
Hello, madam.
Yes, sir.
Madam, I am also one of the co-author in that.
Okay, sir.
Due to that, you can please consider Bannappa sir's comments. It is better.
Sure, sir.
Huh? Okay.
Sure, sir.
Please.
Moving on to next participant. We have Venkateshwar with presenter ID 004.
Hello all, I'm happy to present our work on studying gene mutation in the acquisition of fluoroquinolone resistance in uropathogenic E. coli. Fluoroquinolone treatment forms among one of the basic treatment regimen for anyone with UTI. However, the fluoroquinolones have been found to be ineffective in half of the treated population of patients, and this trend has been observed worldwide. The aspect, women have 50% of lifetime risk of contracting UTI in comparison to 5 to 10% in men. Meta-analysis suggests a recurrence of 20 to 30% in these cases. With this background, we tried to analyze why fluoroquinolones are not effective at present, and the answer to it lied predominantly on antimicrobial. We estimated that the mortality rate by 2050 would be 10 million just because of AMR. As seen in Figure 1, the WHO has categorized organisms under critical, high, and medium priority list. Enterobacteriaceae, a group that reflects E. coli, falls under critical priority list, and thus studies on these are the need of the hour. Figure 2 represents the prevalence rate of various organisms in causing UTI. E. coli tops the list with 82.5% of cases, followed by Staphylococcus saprophyticus with 7.5%. Klebsiella, Pseudomonas, Proteus, and a few other species make up the remaining 10%. As E. coli is the major causative agent, our study focused on E. coli obtained from clinical isolates. With this background, the study design was planned in 4 phases. The first phase was primer design. Second phase included sample collection, characterization, and isolation of DNA. The third phase included PCR assay purification of the product and running agarose analysis. The fourth phase included Sankar sequencing, followed by docking studies. The aim of our study was to evaluate the thyroid gene within the QRDR region of E. coli. Following it was to determine whether any of these mutations play any role in binding efficiencies and characterization of ligands. The receptor in the study would be the fluoroquinolones, and the receptor would be the guide gene. Following IEC approval of clinical isolates, kits were procured for DNA isolation, PCR run, and purification of the product. During primer design, various parameters including potential attachment formation, T-dash complementarity, potential self-limiting site, and PM values needed to be analyzed. As observed under the results section, we see a small extraction from pasta sequence of guide A. Part of the gene highlighted in yellow is QRDR. The helper formation and right flank of this region, primer represented in green. This was followed by sampling and isolation of DNA, optimization and characterization. Table 1 mentions about nanometers. See if the concentration and purity of the isolated DNA is of standard and if it comes out within the kit specified range of 1.6 to 1.9. This is calculated as the ratio of absorbance of sample at 260 rads to 80 nanometers. Sample processing are further evaluated. The figure 4 shows two parts of gel. The first part is basically just uh, binder on plane 1, in a ladder, and on plane 3, we can slightly see the forward timer. The bottom half of the gel is here. The complete gel was run of samples 1, 2, 3, and 4. The biopsy is out below the last fragment of 1 kg DNA ladder, suggesting that sample is 12 to 500 kg. We also performed 32 different sets of docking studies. Figure 5 shows that initially, without resistance, work. And what was the efficiency of binding? This was basically a control. The test would be by introducing mutation. I would finally conclude by saying that considering the rapid development of resistant pattern in common bacteria, studies on genes, uh, so to say, are responsible for resistance and drug uh, moiety modifications will aid in optimization of antimicrobial use. Further, the use of molecular dynamics and the simulation may also play a pivotal role in determining the exact state of binding uh, and various other parameters that need to be accounted for this type of mechanism. Let me take this opportunity to thank MacID for granting us seed money carrying out this work. My sincere thanks also goes to Krupacon College of Pharmacy for uh, organizing this particular conference. My sincere thank is also to MCOPS Mahe for providing us with the state-of-the-art facilities to carry out this type of research work. Thank you, one and all.
Hello.
Hello.
Yeah, that's a very good presentation.
Thank you, sir.
The work which is presented is of a very high relevance. The drug resistance is one of the important the research topic.
Yes, sir.
So, with your findings, what is the outcome of the research relevance to the translational value?
Yes, sir. The simple point is, sir, we have found that mutations with respect to E. coli, it is usually with the DNA gyrase, especially we call it as the QRDR region. That QRDR region, that is quinolone resistance determining region. So when we also analyzed, because there are literature works on it, but not many works have been carried out in India, especially in the southern states. So we also wanted to see whether the modifications or the mutations occur at two basic positions, one at the serine 83 and the other one at aspartate 87. So we wanted to analyze that, and that through our Sanger sequencing analysis, we also have found that other than these two, even at a 93 position, there has been some sort of mutation, but we have not been able to make out what it is exactly. But we are still in the process of continuing the work. Because of the lockdown, the work is still pending, but the literature review and whatever we have done is exactly matching to whatever expected outcome of there.
That's a great findings are no doubt it's going to be helpful.
Yes.
What is the future plan of these findings?
Yes, sir. We have got this work is due to the seed money that we have got from MacID, which is a Manipal, what is that, infectious diseases. We got a 2 lakh grant for this particular work. We are writing for a DBT project, which in the sample size, we're expecting to increase to 500 and above. Right now, we are doing it with lesser samples. Once we get a pilot study done, we are expecting to move up to 500 isolates. and then try to figure out how well we can organize this type of work.
That's great. Okay.
Yes.
Thank you.
Okay. Thank you. Thank you.
Hello.
Yes, sir.
Good afternoon, Venkatesh. And you
Yes,
have
sir.
done
Good
a
afternoon.
good presentation.
Thank you, sir. Thank you.
And it's a need for our actually also the study.
True, sir. True, sir.
Yes. And so fluoroquinolines means you have tried which of the drugs you have
Right
selected?
now we have selected ciprofloxacin, sir. After the advent of nalidixic acid, the use of this particular fluoroquinolones, especially ciprofloxacin, due to its good activity, it has been misused. For which reason there has been resistance of late, especially when we talk in terms of E. coli. I'm more stressing on E. coli because UTI infections, 80 to 85% of the cases are due to E. coli. Yes, sir. For that
and
reason,
even food poisoning also, E. coli is the one of the major.
Very true, sir. Very true, sir.
In
yes,
female
sir.
urinary tract infection, E. coli is the major.
Exactly, sir. Not just resistance, we are also trying to understand the socio-economic impact because women are more affected. 50 to 60% of them are affected in their lifetime. For which reason, yes, sir.
Okay. Then you are taken isolates from which of the patients? Patients isolates or it's
yes, sir. Patients isolate only.
We have that, any categorization you have done means it is collected from which cultures? Usually in the fluid cultures or blood cultures. What
yeah,
type
we have
culture?
urine samples, sir.
It's all are only one kind, means urine samples.
Only urine sample. We are stressing on UTI, so we have just planned for only UTI infection.
One question that when you collected the culture is there is a chance of contamination and there is a chances of other organisms. So how you are identified that isolation, separation and contaminations?
Yes, sir. So that is done by the hospital setup. But after we also got it, they have given us through vials. But once we got through our department, what we have done is we have retested it by EMB agar. EMB is one of the major agar with which we can specifically identify E. coli. Only E. coli, that is a metallic sheen. So by which there were a few, as you said, contamination aspects, which we have discarded in the process.
Okay. If you have a suspect of contamination, you are discarded.
Okay. Yeah, we are directly discarding.
Okay, then you have told a gene mutation. What is the percentage it's present? It is there in your hospital setup. You are expecting is a greater percentage or in the lesser percentage?
Hospital setup, no, sir. We are talking in terms of we have collected isolates which are already showing fluoroquinolone resistance.
Okay. Means almost all patients have resistance cases.
The extreme cases, they have resistance to, yeah.
Okay, in that means this may be the one of the continuation pattern for resistance in future also.
Yes, sir. This is going to be one of the major impact in future also, because we are testing regional aspect right now. South of our coastal district, we are trying to expand it and then go for more samples. Probably that will increase the. We are trying to even understand the genotypic and phenotypic aspects of it.
Yes, good. Okay. Fine, good luck. Thank you for your future indoor research, and hope it will be a very good contribution for present clinical treatment and their changes.
Yes, yes. Thank you. Thank you.
Next, we have Anusha with track ID 006.
Good evening, one and all. I am Anusha. I am from Pharmacology Department. I am here to present the work Combined Efficacy of Decalpis nervosa and Vitamin K2 in Aluminum Chloride-Induced Neurotoxicity, which was carried out under the guidance of Dr. Rajendra Sandu, HOD, Principal, Pharmacology Department, Krupacon College of Pharmacy. Introduction. Neurotoxicity is defined as toxicity within the nervous system that stems from exposure to substances known as neurotoxins. In our daily life, we are exposed to neurotoxicants through several sources such as food, drugs, heavy metal, cosmetics, environmental factors, and occupational factors. In this study, aluminum is used as neurotoxicant, which has its mechanism through generating the oxygenated free radicals and oxidative stress, causing neurodegeneration. Decalpis nervosa and vitamin K2 are used for treatment. Decalpis nervosa contain alkaloids, polyphenols, flavonoids, carbohydrates, and other active principles. These possess antioxidant activity and reduces the oxidative stress. And vitamin K2 also has, and their derivative, they protect the neurons from oxidative stress. So keeping all this in view, the current study is planned and carried out. Materials and methods. Preparation of plant extract. It was prepared by maceration, 70% ethanol, and the yield was found to be 5.45%. Phytochemical analysis was carried out. GCMS analysis was carried out. In vitro antioxidant test DPPH assay was conducted. Induction of oxidative stress, neurotoxicity, and evaluation of neuroprotective activity. 30 male albino visitor rats were randomly assigned for 5 groups containing 6 animals each. First group, the normal control was given corn oil. Second group, positive control was given with aluminum chloride, 100 mg per kg. And the third, fourth, fifth group were taken as treatment. Third group was given with extract Decalpis nervosa, 500 mg per kg. Fourth group was given with vitamin K2, 100 microgram per kg. Fifth group was given with extract and vitamin K2 in combination. Behavioral assessment, actophotometer. Motor activity was measured by placing the animal in the actophotometer. The walking and running activity was measured. Elevated plasmase test. The animal was placed in one end of the arm, and the time taken for it to move from the open arm to closed arm was recorded as initial transfer latency, and this was repeated after 24 hours. A test for condition avoidance response. The cognitive function was assessed by using Cook's pole climbing method, and every rat was subjected to maximum of 10 trials per day. And when they gave 3 consecutive avoidance responses, the training was completed, and the cognitive function was assessed on 42nd day of aluminum chloride dosing in respective groups. Biochemical studies. Aluminium levels, acetylcholinesterase enzyme level, dopamine, serotonin, and total protein was estimated. Oxidative stress markers, T-bars, catalase, superoxide assays were carried out in brain regions. Results, actophotometer, the aluminum chloride group, which had less locomotor activity, and the other group had significantly increased the locomotor activities. Plasmase, the group which was induced with aluminum chloride has increased transfer latency, which was significantly decreased by the treatment. Test for memory, aluminum chloride increased group showed increased escape latency, and this was decreased significantly with the treatment. Aluminum levels and acetylcholinesterase levels were increased in aluminum chloride induced groups, and this was significantly decreased by the treatment. Dopamine and serotonin levels were decreased in the aluminum chloride induced groups, and which were increased by the treatment groups, Decalpis nervosa and vitamin K2. Neuroprotective cytochrome by GCMS analysis were vanillin, alpha-amyrin, oleic acid, betulin, L-ascorbic acid, 2,6-dihexadecanolate. Phytochemical estimation showed tannins, flavonoids, carbohydrates, alkaloids, proteins, steroids. Antioxidant parameters, SOD catalase were increased, T-bars were decreased in the treatment. Discussion. Aluminum chloride administration, it significantly decreased the locomotor activity, distorted cognitive function, produced learning impairment, and this was improved by the vitamin K2 and Decalpis nervosa treatment. And the observed neurochemical neurobehavioral impairment may also be due to aluminum-induced plaques and tangles in rat brain. Interference with cholinergic function and exacerbation of inflammatory process showed that how aluminum would cause learning and memory deficit. It also showed decreased cholinergic neurons, increased acetylcholinesterase enzyme levels, and decreased 5-HT levels, dopamine levels, and increased the oxidative stress biomarkers. So ability of Decalpis nervosa to inhibit all these activities were presence of flavonoids, phenolic compounds, fatty acid, which were reported by GCMS study. To conclude, Decalpis nervosa as well as vitamin K2, they possess a potent neuroprotective activity, and due to the presence of antioxidant principle. Further estimation of different molecular markers is required to prove the exact mechanism of Decalpis nervosa as a neuroprotective compound. These are few references. Thank you.
Anusha, you can switch on the video. It's open for question answer session.
Anusha, very
Yes,
good
sir.
work.
Good afternoon, sir.
Good afternoon. Very good presentation. Uh, very nice work. Uh, and I would like to know that is there any traditional uses for this Decalapis nervosa?
Sir, in the literature found that the other species also they had like antioxidant therapeutic use, like antioxidant property. So it took this work, sir.
Yeah, that's right. Exactly, the Decalapis hamiltoni
Yes, sir.
is a one traditionally used medicine for this neurological disorders. So I was just surprised that where from this Decalapis nervosa you brought it, and what is its traditional claims, if any?
Sir, it was given, this idea was given from a cognitive department teacher. So then he had only few amount of drugs. Then we procured from the vendors and we bought it, sir.
Okay. What is the translation value? Is it easily available and can be scaled up?
This when we bought, yes, sir, the vendor has given it not to. It's not completely easily available because like so many species of other the plant are like, endangered. It's not completely
Exactly.
easily. Yes, sir.
Exactly. That's what my concern is actually.
Mm.
It's an already a rare endemic plant. So it is endangered already. So the plants which are red listed, if you do any the studies also, actually it will be harmful for the environment. It's not a sustainable approach.
Okay, sir.
Okay. So anyway, the good work for your learning, it is okay.
Mm.
My concern is about its translational value.
Yes,
Okay.
I understand.
Thank you.
Thank you, sir.
Hello.
Good afternoon, sir.
Good afternoon.
Good afternoon, sir.
Already Banappa sir asked so many questions. So I
Yes,
have
sir.
concerned with one methodology.
Yes, sir.
You have done what are the parameters? Just briefly tell me.
Behavioral assessment, actophotometer, elevated freshness.
So in behavioral, you have whether you have trained the animals
Yes, sir.
before giving the aluminum chloride.
Yes, sir. They were trained, and after giving aluminum chloride and then treatment, then it was assessed again, sir.
Okay. How many days you have done training?
Training was done for 10 days, sir.
10 days. Okay. After that, how you have selected the animals?
Then it was which had a better response, like with that actophotometer. And only those animals were selected, and then they randomly assigned for groups, sir.
No. Better in the sense you do not have any some parameter you measured this much of readings you come you will select, and if it is this much of reading is comes, so you will be rejected. You have not done any particular parameters. Means
We trained it, sir. Like animals in that we trained it, but we didn't count to that numbers.
That is very important, especially in this kind of studies.
Okay, sir.
Where some of the animals will show the response, some of the animals will not. The selection of animals will be very important.
Yes, sir. The one which showed the response that was selected, sir.
Okay.
But we didn't keep it in a number.
Okay.
Thank you, sir.
So aluminum chloride will be the main mechanism is mainly is by free radical mechanisms.
Yes, sir.
It is probable. Then you tell me why you are selected, how it is get contaminated to human, how it is causes the possible neurotoxicity, how you are related to these studies to human. What is a possible cause for neurotoxicity in human?
Sir,
what are the sources of aluminum chloride?
Yeah, for the purification of water it is used. Then through any other environmental this it can be ingested by two humans.
That it is very important that you please go through that. Okay.
Yes, sir.
If it is stored in aluminum container, there is a leach of aluminum with chloride, and it becomes aluminum chloride to water and all. That is important. Okay, no, that's you just go through that. That is otherwise, work is good, effort is good, and as I told you, selection of plant, I have completely agreed with Banappa sir. So this is very important. But other than that, is good. Okay.
Thank you, sir.
So we move on to the next participants. It is Charitra. Participant ID is T1PCOL005.
Myself Chaitra from Palladium College of Pharmacy, pursuing my MPharm in the Department of Pharmacology. I'm here to present the work on effect of Maduka longifloria bark extract against experimentally induced rheumatoid arthritis in rats, which was carried out under the guidance of Mrs. Jyoti Bai, Assistant Professor, Krupacon College of Pharmacy. So now, first, let us start with the introduction. Rheumatoid comes from rheumatism, which broadly refers to musculoskeletal illness. Rheumatoid arthritis is a chronic systemic autoimmune condition associated with symmetric synovial joint inflammation that may result in progressive joint damage, bone destruction, and also affect adjacent organs and tissues. It is explained as a widespread connective tissue disease that affects para-articular structures such as tendons, bursae, tendon sheath, extra-articular tissue such as subcutes, etc. It usually occurs in female at the age of 50 to 60. In the current study, the plant Maduka longifolia is used to investigate its usage in rheumatoid arthritis. It is indicated in the literature that chemical constituents like oleanic acid, alpha-spinosterol, alpha-amidine acetate, etc. are acting as an anti-rheumatoid arthritic. So, keeping all this in mind, the present study was carried out. First is materials and methods. In this, preparation of plant extract. The plant was collected, dried, and extracted by Soxhlet apparatus using 70% ethanol, and the extract was dried and used for experimental purpose. Next, phytochemical analysis. Preliminary phytochemical analysis was carried out, and it was seen that the presence of alkaloids, carbohydrates, phytosterols, proteins, saponins, flavonoids, tannins, etc. Experimental models. 30 female albino rats weighing of 180 to 200 grams were randomized based on their body weight into 5 groups of 6 animals each. Group 1 is taken as control group, which is treated with normal diet. Group 2 is taken as positive group, which is treated with CFA. Group 3 is taken as low dose, that is treatment group, low dose of extract with complete fluent adjuvant. Group 4, high dose of plant extract with complete fluent adjuvant. Group 5 is taken as standard group, which is treated with CFA and ibuprofen. So this experiment was carried for about 22 days, and the following parameters were established. That is, physical parameters. Body weight per volume was measured from 0 to 22nd day. Motor coordination test was carried out using Rotarod apparatus. Hematological parameters. Hematological parameters is carried out by taking the rats. They were anesthetized. The blood was connected from retroorbital sinuses. The RBC, WBC, and platelet was estimated. Radiographic analysis. Rats were taken to X-ray center. X-ray images were taken for the affected paw of experimental animals. Next, histopathological parameters. At the end of studies, all the rats were sacrificed by excess anesthesia and tibiotarsal joints were collected from each group, washed in ice-cold saline, and subjected for histopathological studies. Results. Body weight was compared between the positive treatment and standard group. The positive showed less gain of weight as compared to the standard and treatment group. Per volume, there was increase in per volume of positive group as compared to the treatment and standard group. Motor coordination, there was decrease in fall of time as compared to the treatment and standard group. Next is radiographic analysis. Radiographic analysis, mainly the tibiotarsal knee joint is taken. It is shown that there is swelling of soft tissue and narrowing of the joints in case of CFA induced as compared to the control, high treatment, and standard. Next, we have histopathology. Histopathological studies of tibiotarsal joints showed destruction of cartilage, erosion of osteoid, inflammatory cells, and increased vascularity in adjacent cells in CFA-treated animals. Maduka longifolia treated rats produced joint protection compared to arthritic rat by reducing destruction of cartilage. So now the present study was initiated to investigate whether Maduka longifolia bark extract could offer anti-rheumatoid activity against complete fluence adjuvant induced rheumatoid arthritis with lesser side effects. In the present study, anti-rheumatic effect of Maduka longifolia was evaluated mainly by in vivo study. CFA treated rats showed less body weight gain, increased paw volume, and decreased fall of time as compared to ibuprofen and Maduka longifolia extract. Radiographic changes in rheumatoid arthritis conditions are useful diagnostic measure which indicate severity of the disease. Maduka longifolia bark extract group receiving at the dose of 60 mg per kg and ibuprofen show significant prevention against bony destruction, showing less soft tissue and swelling and narrowing of joint spaces. The present study demonstrated that treatment with Maduka longifolia bark extract, ibuprofen significantly potentiated anti-rheumatoid arthritis activity against complete fluent adjuvant-induced rheumatoid arthritis. And these are few references. Thank you.
Now it's open for question answer session.
Chaitra?
Yes, sir.
So again, this is also one of the important area where there is no effective medicine available, particularly for the rheumatoid arthritis. It's a good work, definitely, and the findings are very promising. In fact, it has reduced the all the key parameters associated with rheumatoid arthritis. What is the rationale behind your selection of this plant?
Actually, traditionally, it is used mainly as analgesic, sir, and anti-inflammatory also. The oil from this bark and all, it's used for analgesic, antipyretic, and even for anti-inflammation. And searching all the reviews based on this, this plant was selected. And this work was not done on this, mainly the bark part. So I have chosen this.
That's what I have these two questions. One is in which system it is being used, and what is the part which they have used? Because the bark is whether is a sustainable approach. If you find any effective results, also whether it can be translated.
Yes, sir. Bark, we can extract it and use it for that purpose.
No, you can.
Usually, they extract the oil from that and use it.
Okay. So you said that it is traditionally used.
Hmm.
In which traditional system of medicine it is mentioned?
Like in Ayurvedic, sir. Usually, normal tradition also. We also sometimes take that oil and use for many of pains, and even if we have any upset in the stomach, for that also we use it.
Okay. Which part of the country it is routinely used?
In our country, it is used, sir.
No, which part? No, because would like to know.
Northern part of India, mainly it is available more.
Okay. Maduka indica. Okay. All the best.
Thank you, sir.
May I have one more question?
Yes, sir.
Chaitra,
Yes, sir.
what are the chemical compounds which are responsible for this activity?
Like oleanic acid, sir, alpha spinosterol, which was alpha spinosterol, alpha amyrine, and erythrodiol, monocaprylate, flavonoids, all these chemical constituents, they were extracted. They were seen to give this anti-arthritic activity.
No, in your study, is there any correlation between that specific compounds? So do you attribute that all the observed effects are because of any compounds in that?
Because these constituents are present, only then it is showing that activity, right? So keeping that in mind, we have given this.
Okay, good. Okay, carry on. All the best.
Okay.
Chandrasekhar sir, over to you.
Okay. I have only two questions. One,
Yes,
why you
sir.
selected ibuprofen as a standard drug?
Because usually ibuprofen is used to reduce pain, inflammation. Like usually doctors also prescribe this many times. So keeping that, and also they are searching the research articles on this ibuprofen was selected.
Okay, otherwise your work is good, and there's no much this thing you can further scope. You can measure some histamine and interleukins and some parameters. They are also contributing factor for that if you want to continue for future. Okay.
Okay, sir. Okay, sir. Sure.
Okay. Okay. Good luck. Thank you.
Thank you, sir.
Chaitra, one more
Sir.
question.
Yes, sir.
For you. So, which method of extraction you have used?
Soxlate, sir.
Soxlate. And you are claiming the effect due to these essential oils?
Sir, I didn't get.
The chemical component which you have mentioned is in the essential oils, no? Does you get in the Soxlate method an essential oils getting extracted?
Not the bark. From the bark, the crude product is extracted by Soxlate, sir.
So the active constituents may not be the essential oils because while you extract with
No.
What is the solvent used?
Ethanol.
So you just cross check because it is less feasible with there's an alcoholic the oxalate method. So getting these oils in the form of active constituents.
Okay, sir. Sure.
Thank you, sir. Moving on to the next participant. It is Hemant. It's track that is identification number is T1PCOL007.
Good afternoon to my teachers, expertise, and my colleagues. Today I am going to present my dissertation on topic anti-ulcer activity of Mutinja calibra field against experimentally induced gastric ulcer in rats, which was under the guidance of Jyoti White. Let's start from the introduction. The most common ulcer within gastrointestinal tract is septic ulcer, usually because of hyperacidity and less production of cytoprotective mucus, which causes erosion in mucosa equal to or greater than 0.5 centimeters. Recently, lots of chemicals or allopathic drugs are available in market for the treatment of gastric ulcers, such as proton pump inhibitors, acid blockers, antacids, etc. Because of several side effects of synthetic medicine, nowadays many researchers are going on to discover natural alternatives for treatment of that problem. Mutinja calibra is the plant which we took for research with belongs to Pilocarpaceae family found easily in both sides. The flower is white in color, whereas ripened fruit is red in color and unripe fruit is green. This plant is well known for its anti-inflammatory and anti-platelet properties. Based on the literature survey, the chemicals which are responsible for the treatment of gastric ulcer are flavonoids, tannins, and phenolic compounds. Materials and methods used for our research is the chemical constituent for the plant was extracted by use of methanol, and for our this study we need some gastric ulcer induced rat. So gastric ulcer was induced in rat by three methods, that is pylorus ligation, methylene blue induced, and cold water immersion stress induced gastric ulcer. The results from our research is oral administration of methanolic extract, that is 200 milligrams per kg, should significantly reduction of gastric ulcer when induced by cold immersion induced and methylene blue rat compared to pylorus ligation. The effects of Mutantia calabra fruit extract was compared with standard drug, that is, pantoprazole in the dose of 20 milligrams per kg. When comparing ulcer score in all these three ulcer models, it was found that 200 milligrams per kg methanolic extract of Mutantia calabra fruit was more effective in cold water immersion model compared to methylene blue and pylorus glycation model. Discussion. The main aim of anti-ulcer drugs is to reduce acid secretion as well as to increase the mucus secretion in stomach. So the results from our study prove that high dose, that is 200 milligrams per kg of Mutantia calabra fruit extract, reduces the acid secretion as well as increased protein level, resulting in gastric cytoprotection in rat. So we conclude that from the findings of our study, it suggested that Mutantia calabra fruit exerts its gastroprotective activities via several mechanisms of action, and it causes an indirect effect on release of gastric hydrochloric acid and protects gastric mucosal damage. And these observations activities may be attributed to phytochemicals present in them. And these are the references which we use or which we followed for our research studies. Thank you for granting me an opportunity to present this thesis. Thank you.
Now it's open for question and answer session.
Good afternoon, sir. Good afternoon, sir.
Good afternoon, Hemant. It's a good work, definitely. So what is the method of extraction you adopted?
Oxalate extraction, sir.
Sorry?
Use methanol.
Methanolic oxalate apparatus.
Yes, sir.
Okay, okay. That's all. Okay.
Hello.
Yeah, good afternoon, sir.
So you are selected the plant for anti-ulcer activity.
Yes, sir.
So what is the probable mechanism of your plant? How it is reduced the?
It consists of main active constituents such as flavonoids, phenolic compounds, and tannins, which are responsible for the treatment of gastric ulcers.
No, you don't know mechanism.
Mechanism
how they are neutralized
by reducing the gastric acid secretion and increasing the mucus secretion.
Okay, fine. Good work and good luck. Okay.
Thank you, sir.
Next, we have Miss Archana with track ID 008.
From Nandaka College of Pharmacy. My presentation is about evaluation of anti-cancer effect of using in mice. In this study, using effect is observed on a DNA and humanoid-induced cancer usually is known to occur due to the development of abnormal cells, which is known to have the ability to attack or spread to various body parts. DNU is known to act on the nuclear factor of T cells and caspase-3, and is known to activate the inflammation and apoptosis factor, which leads to the formation of skin cancer. Proton will act on the PGE2 and results in the DNA and polyamide synthesis and activator protein 1 activation, which leads to the formation of the initiated cancer cells caused by the DNU. The active toxicity study on using is obtained by various research articles, and thus the dose of 12.5 mg per kg is selected. Methodology. Swiss albino mice of 10 to 12 weeks old weighing around 25 to 35 grams are selected and divided into 5 groups. Group 1 control group receives double distilled water up to 16 weeks. Group 2 disease control group receives single application of PMDA and propanol is applied 3 times a week topically up to 16 weeks. Group 3 topical group receives glucine applied 2 weeks before doing the application. application period. Group 4 oral group using received 12.5 per kg orally for 2 weeks for DMT application. After 2 weeks, single application of DMTA and propanol is applied topically 3 times a week up to 16 weeks. Throughout the experimentation period, using is given orally. Group 5 oral plus topical group using drug is applied and given orally for 2 weeks. After 2 weeks, DMT application and propanol is applied 3 times a week up to 16 weeks. Throughout the experimentation period, using drug is given orally and topical application is conducted throughout the experimentation period. Parameters which are conducted to study the effects of drug are body weight, tumor incidence, cumulative number of tumor average latent period, and histopathology studies and biochemical parameters are also conducted after the termination of the experiment. Biochemical parameters are SOD, catalase assay, lipid purification assay were analyzed. Results, the physical parameters, that is body weight, cumulative number of tumors, tumor incidence, tumor yield, and average latent period have been plotted below with this graph. Body weight, the body weight has been plotted and it shows that the body weight of the disease control group is gradually decreased when compared with that of other test groups. The tumor incidence cumulative number of tumors, tumor yield show that the tumor in the disease control group is more when compared with that of the test group. Average latent period is nothing but a lag between the application of the morphine agent and appearance by 50% of tumors. Thus, the result shows that the test drug delays the initiation of the tumor when compared with that of the disease control. Biochemical parameters, lipid peroxidation assay, catalase assay, and SOD assay is conducted. Lipid peroxidation is the oxidative damage that affects the cellular membrane, lipid proteins, and other molecules leading to their damage. This result shows that the damage in the disease control group is more when compared with that of the test group. What do you want? SOD and catalase assay. Catalase assay is a biochemical test which detects the production of catalase enzyme in organism, which is known to protect the cell from oxidative damage, and thus the damage in the disease control group or the catalase enzyme in the disease control group is less when compared with that of the test drugs. And SOD assay. SOD assay is enzyme which is known to provide cellular defense against reactive oxygen species. Thus, the SOD enzyme is less when compared with that of the test groups. Histopathology studies. First is normal skin histology. The group 2 shows epidermal hyperplasia, hyperproliferative epidermis, and hyperkeratinization. Group 3 shows a slight formation of keratin pearls, whereas group 4 shows a decrease in thickening of epidermal layer, that is, epidermal hyperplasia is less. Group 5 shows that tissue was observed to have a slight abnormal structures on the epidermal layer. Thus, this studies shows that the test drug Krupacon showed a significant anti-cancer effect in animals against the DNA and propanol-induced skin cancer in mice. Conclusion: from the result obtained, it was evident that the drug Krupacon was a significant effect against DNA and propanol-induced skin cancer in mice.
Evaluators. The panel is open for question answers.
Dr. Chandrasekhar sir.
Really, sir.
If you have any questions, you can proceed, sir. I don't have anything.
So you have selected this plant.
Yes,
so how you have selected this plant for this anti-cancerous activity? Do you have any rationality?
Sir, this drug was used for hepatocellular carcinoma and also in anti-inflammatory and analgesic activity.
Already it is proved. Okay.
Yes, for those it is proved. For skin cancer, it was not done. So on that basis, I took this drug for the skin cancer purpose.
Selected for this. Okay.
Yes.
Okay. Good luck.
Thank you, sir.
And nice work. You are done.
Yes, sir. Thank you.
Next, we have Rashmi. with track ID 010.
Models have long been used to investigate and characterize disease pathogenesis. Good afternoon, everyone. My name is Rashmi Nirvati. I'm pursuing second year MPharm in Department of Pharmacology, Daily College of Pharmacy, Mumbai. My track ID is T1PCOL010. The title of the topic I have chosen for this poster presentation is Experimental Induction of Lactic Acidosis. Animal models have long been used to investigate and characterize disease pathogenesis, target identification, as well as to evaluate new therapeutic agents. Metformin and other biguanides are commonly used to treat type 2 diabetes mellitus. Although very safe, they too are riddled with a major side effect such as lactic acidosis upon chronic usage. Lactic acidosis is a life-threatening condition marked by low arterial pH and high lactic levels in the blood. Primarily, its prevention and early treatment are therefore required to minimize post-lactic acidosis consequences and associated death. There are very far and few experimental models to mimic the lactic acidosis. Further, the limited efficacy of existing treatments for lactic acidosis is a challenge. Hence, the development of new experimental models and therapeutic approaches are needed. The objective of this study is to review the existing animal models for lactic acidosis. Mainly, there are two types, chemical induction and chemical induction in genetically modified animals. First one is fenformin-induced lactic acidosis in viscera rats at a dose of 64 mg per kg per day given intravenously for 15 days. Here are the outcomes. A reproducible model of lactic acidosis was obtained by the combination of experimental renal failure and treatment with fenformin or pharmacological use. Second one is metformin, metformin, and fenformin-induced lactic acidosis at various doses in straw dolly rats given intravenously for 4 hours. The severity of lactic acidosis induced was higher after each dose of fenformin, followed by metformin and metformin. Third one is fenformin and metformin-induced lactic acidosis in F344 JCL rats at a dose of 100 to 400 mg per kg per day of fenformin and 50 to 200 mg per kg per day of metformin given orally for 28 days. In this study, metformin did not lead to change in the lactic acid levels in rats equivalent dose to human therapeutic exposure, while fenformin-induced persistent elevation of lactic acid levels. This is the last one, that is genetically modified model. Metformin-induced lactic acidosis in OCT1, that is organic cation transporter 1, and wild type FPB mice at a dose of 150 mg per hour per kg given intravenously for 3.5 hours. The blood lactate concentration in metformin treated wild type mice was 2.5-fold greater than that in metformin treated OCT1 mice. The plasma concentration trying profiles of metformin were similar in wild type and OCT1 mice. The conclusion of the study that this work briefly illustrates the challenges associated with experimental induction of lactic acidosis that could lead to help the preclinical assessments and identification of therapeutic approaches for translational application in humans. By comparing all these four different models, cost effectiveness wise, metformin induced lactic acidosis in rats is effective for chronic use. Otherwise, the genetically modified animals can be used for short-term treatment. Thank you.
Out of which model is highly feasible in laboratory, easy and affordable and reproducible model?
Fenformin is banned, so we are approaching metformin-induced lactic acidosis model.
Okay.
Rashmi?
Yes, sir. Good afternoon, sir.
Where this work was carried out?
Sir, I have reviewed 4 papers. Currently, my research work is on lactic acidosis model only. So it is still in process.
Okay. Okay.
Okay, thank you, sir.
Over to the moderators for the next presentation.
Okay, sir. Next we have Mr. Basir with presenter ID 011.
Hello everyone, my name is Mr. Bashir. I'm from Alumni College of Pharmacy. I'm going to present a poster entitled Impact of Oxidative Stress and Neuroinflammation in Parkinson's Disease, a review. So neurodegenerative diseases such as Parkinson's disease and Alzheimer's disease impose a pressing burden on our developed and consequently aging society. Parkinson's disease is a progressively debilitating neurological disorder that affects about 6 million people around the world. This disease is mainly characterized by the progressive and irreversible degeneration of dopaminergic neurons localized at the substantia nigra compacta on the mesenteric pylon. Reactive oxygen species, free radicals, and misfolded protein aggregates are the critical features of all the neurodegenerative diseases, but the specific role in Parkinson's disease and neuronal cell death remain unanswered. Lately, its oxidative stress and neuroinflammation process were recognized to play a key role in the pathogenesis of the Parkinson's disease. Various preclinical studies, PET scanning, brain imaging, and postmortem studies involve innate and adaptive immunity in neurodegeneration. The subject of the research was to identify whether the oxidative stress and inflammatory processes are involved in the etiopathosis of Parkinson's disease. The role of the alpha-synuclein in progression of the Parkinson's disease is studied. In this review, we describe the concept of involvement of oxidative stress and neuroinflammation in Parkinson's disease associated neurodegeneration. So we will see basically the mitochondrial of mitochondria in this. So the key source of oxidative stress involved in pathogenesis of the Parkinson's disease is mitochondrial dysfunction. The superoxide radicals are produced in the mitochondria as byproducts. During oxidative phosphorylation, the leakage of the electrons leading to the Auror's generation is caused by the damage to the mitochondrial complex 1 in electron transport chain. The dopaminergic neurons are more vulnerable, intrinsically more reactive oxygen species generating. MPTP and troponin being the mitochondrial complex 1 inhibitors when given intraperitoneally exerts dopaminergic cytotoxicity. Moreover, the reduction in the activity of the mitochondrial complex 1 has been observed in tissues of the patients with Parkinson's. The genetic mutations of the mitochondrial proteins, sparking BJ1 and pink earlier forms of the Parkinson's disease condition and evidence for mitochondrial dysfunction related to the dopaminergic cell damage and oxidative stress. The reduced respiratory activity along with the oxidative stress has been seen in the rats representing Parkinson's. So, the inflammation, regarding the inflammation, misfolded protein aggregates are a critical aspect of several neurodegenerative disease. Nevertheless, several questions remain unanswered regarding the role of misfolded protein aggregates in the cause of neuronal cell death. Recently, it has been postulated that neuroinflammatory processes that play a crucial role in the pathogenesis of numerous demonstrated linking the oxidative stress and the neuroinflammation to neurodegeneration. So it was seen that in post-mortem, what we got in blood and CSF samples in positron emission tomography in risk factor studies were done comprising of the genetic studies and epidemiological studies. Neurotoxin test models was the evidences were taken out from that. Immunomodulating strategies, viral vector-based and Sinoprin transgenic models were seen. All the evidence were collected, which I put forth in the poster one by one. What was seen increased elevated levels and what was seen the decreased elevated decreased levels. What was seen the increased pro-inflammatory was seen in some in blood and CSF samples and like. So in our conclusion, what we got to know about this poster is that neurodegenerative disorders threaten our society with a substantial economic burden. This is an urgent need. There's an urgent need to develop an novel therapeutic strategies taking underlying disease pathogenesis in order to slow down or halt disease progression. In this regard, it is of utmost importance to better understand how neuroinflammation and oxidative stress plays a role in the initiation progression. Animal models, human studies are generating multiple evidences for the involvement of microglia and T lymphocytes in EPD. However, a specific role in disease initiation progression remains elusive. So the recruitment of the peripheral monocytes has also been able to contribute to PD-associated neurodegeneration. But the exact role of these peripheral monocytes in the disease remains yet to be known. So further, research should focus on identifying new drug targets by broadening our understanding of neuroinflammatory process in PD-associated disease, initiation and progression.
The panel is open for question answer round.
Yeah. The oxidative stress, neuroinflammation is the crucial pathological consequences which are playing role in this disease. Good attempt, but I was unable to see the slide and hear you properly. Anyway, I appreciate your effort. Congratulations.
Good afternoon, sir. Thank you so much.
Hello.
Hello, sir. Good afternoon.
Can you tell me within fraction fast which is advances research treatment is taking place in Parkinsonism? You have done an extensive review of literature, no?
Yes, sir. Yes, sir. So from this neurodegenerative disease, it's a basically threat to our society. For
No, just you tell me point to point. Please point to treatment only. Okay.
yes. So right now we don't have a particular treatment for these neurodegenerative disease, be it Parkinson's disease or be it Alzheimer's disease. Right now, in present scenario, what we have is the symptomatic treatment. And so we don't have a basic how to since we don't know exactly the cause of these neuronal cell death, the perfect treatment is yet to be known. And right now in present scenario we only have symptomatological treatment, sir.
Okay, fine. Good luck. Huh? Thank you.
Thank you so much.
Okay, nice.
Thank you so much.
Moving on to the next participant, we have Farmiza with poster ID 013.
Today I am here to present one of the research work. We have been considering accounting for diabetic care study in diabetic model in this program. The following order is Dr. Rekha. Coming to the diabetic has become a critical clinical issue affecting more than 482 million people worldwide, and the number is supposed to be increased approximately by 552 million by the end of 2020. More than 78 million approximately $8,669 have been spent on treatment per patient every year. Studies have suggested that a lower limb is being amputated for every 30 seconds because of diabetes. 85% of the amputations in diabetic patients are due to food alteration, which may eventually be related to severe gangrene and infection. Qualitative options available. The current alternatives are being expensive and consuming and they are dealing only with the prevention of infection. Very less studies are also being conducted. So, in for this purpose, we have selected this study and we have selected a new system which will be hydrogen, which will show less side effects because the marketing compounds are showing carcinogenicity in the case of long exposure, and here will be hydrogen, which is a natural compound obtained from ginger. It is a half analog of curcumin. It is activity, anti-inflammatory activity, antimicrobial activity, cytotoxic hormones, cytotoxic activity, and also has antioxidant properties. Initially, the healing activity which has been doing in the longer use is reported, but its mechanism and its activity in the hypnotized idiopathic symptoms are explored yet. We are thinking that it may be because of its promising antioxidant properties, it may help in the healing of the wound. In the method, we have divided our animals into three groups: trauma control, heat control, and case test, which is 100 mg per kg. Oral glucose tolerance test is also performed. This oral glucose tolerance was performed in order to find out whether we have achieved or insulin resistance or not. So basically, here we are such type of reminders which can measure type 2 diabetes in women. So in order to create the type 2 diabetes as well as hyperglycemia. So insulin resistance is being achieved by giving high fat diet. Here we have given high fat diet for 5 months, followed by low dose of cephalosporin. So first we want to find out whether our animals have achieved resistance or not. Further, we have wanted over glucose tolerance here. Here, high fat diet and normal diet fat and for taken, and they have been taken from a very low age of 4 years. At the end of 5 months, we have been feeding them high fat diet. At the end of 6 months, we have detected blood glucose levels in the measure, and here we can see this graph that the normal diet fat rat from the 15th moment, they have been that and then they are but then compared to the high fat diet has been reduced till the end of 120 years. So by this we have found out that our animals have achieved insulin resistance. Then we have gone to induce hyperglycemia using low dose of cephalosporin via IV. So after delivery of that cephalosporin, we have left the animals for stabilization up to 1 month. After 1 month, we have tested the glucose levels, and also we have gone to the private life total cholesterol, HDL, LDL levels in order to confirm that our randomization was based on the particular thing that we need to because we want to create chronic blood glucose levels. So we want to create a group where chronic blood glucose levels are present. So that's why we need to have randomized only those animals whose blood glucose levels are greater than 20 or more than that, and other animals. So here we can see that cardiovascular and LDL of the normal animals are less than compared to the disease control animals. So that indicates that animals have achieved type 2 diabetes with both insulin resistance and an indicates that they are confirmed or glucose tolerance and hyperglycemia has been achieved that we have confirmed by the glucose profile test. And here you can see that HDL is less than compared to the normal. That is like in diabetic patients, triglycerides, cholesterol, as well as LDL are So basically here, because of this has a pattern, so we need to take up to 15 days or more than that. But we have selected day 5, day 10, and day 15. So we have compared to normal disease and we have taken so here we have selected identical feature and the overall hepatoviral was given regularly at the end of the 15th day. So from the day 0, we have been given the overall hepatoviral in 5 days. Then that particular set of animals of day 5 were being sacrificed at the end of the day 5 and then their bone area was measured, the percentage of sugar was measured and then the rest of the bone tissue was analyzed to pathological study. Similarly, the day 10 group as well as the day 15 group of animals were continued then oral research is involved. So at the end of the day 10, we have been again sacrificed the animals and take area and group and percentage of protein was calculated. Again, for the 15 set of animals, we have one set of animals here we can see that the blue contraction area and the at the end of 15 days. Here the percentage is going to be also measured. Significant group was seen in the tested group at the 100 per kg compared to the normal. So we have done the day 10, we have seen the ulcerative epithelium in the normal individuals as well as the test group. We have seen that there are the proliferative fibroblasts and then here we can see the convolution tissue also. But when compared to the day 10, we can see the slide of day 15. Here, the picture is showing highly ulcerative epithelium that scab containing cell disease. Abundance of neutrophils can also be seen. Extravascular adhesion fibrosis is also seen. Showing the area the proliferation of fibrosis. Here we can see both of these also. The start area of the compared to the here the start area is here we can see that here the ulcerative epithelium is present. But here we are not able to see the the granulation tissue here we can see the granulation tissue and here this is the granulation tissue. But here we will see the granulation tissue found and young fibroblasts present, young neutrophils were present here. Purple collecting the neutrophils and here some fibers I think they are present with they are fibroblasts. These are very low in reduced compared to normal. Apart from that, the collagen has been formed and the linkage is also seen in the metabolism and normal control compared to so in conclusion, we have made that sympathy metabolism has been effective in treating the diabetic when compared to the normal metabolic. So our drug has shown a promising timing to the management of diabetic. Further research is going on. We are finding out by which mechanism exactly our drug what is so these are the references we have followed. And the oral study and the patient, the medical college, and the ALC provided the therapeutic and the fellowship.
Chandrasekhar sir.
Hello.
Chandrasekhar sir, you can proceed with the questions, sir, if you have something.
Okay. Hello. I want one question that how the lipid profile, what is the correlation of lipid profile with the wound healing activity?
Sir, basically we have yes, sir. Basically, we have randomized only on the basis of this lipid profile only, sir. So if animals' blood glucose levels are more than 300 mg per dl, so and also in case of diabetic people, this lipid profile like triglycerides, LDL, as well as this total cholesterol, it is high, sir. So that's why we have taken these things into consideration.
Okay, that is what my question. How
Yes.
they influence the significance of high level of triglyceride and cholesterol in wound healing activity? What is the mechanism?
So basically, this here we have just like in order to confirm that it is having diabetes or not, we have just taken this into consideration, sir. But we have maximum take consideration of fasting the glucose levels only. But this is like in order to confirm that they are having diabetes or not. That's why, sir. So basically, there is a
Okay. Okay, fine. Good.
okay, sir.
Okay.
Thank you, sir.
Next, we have Sandeep with track ID 012.
Good morning. Myself Sandeep Chandkote. I'm from BLDSSM College of Pharmacy, Vijaypur. My research title is Evaluation of Neuroprotective Property of Citrus Pulp Powder in Scopolamine-induced zebrafish model amnesia. The abstract is the present work is evaluated the neuroprotective property of citrus pulp powder at a different doses 50, 100, 200 using the scopolamine-induced zebrafish model. So in our present study, the neuroprotective activity was assessed using a novel tank test of swimming behavior recorded. So here the administration of sample was shown the significant increase in the number of entry on the top and number of entry number of the time spent in the top, and the significantly decreases in the number of entry at the bottom and the time spent in the bottom. We also evaluate the biochemical parameters like acetylcholine and MDA. The treatment of citrus pulp powder at a different doses has shown the decrease in the acetylcholine and as well as MDA. So introduction, the Alzheimer's disease is a neurodegenerative disease. So it is associated with age-related, and the characterization is memory loss and dementia. Due to the Alzheimer's disease, the loss or the progressive loss of cognitive abilities as well as the behavioral disturbance. The zebrafish model was assessed already in some more literature, and it also used for the in vivo model to induce the Alzheimer's disease for the screening model, for the screening. And the citrus pulp powder or citrus lemon is evergreen tree, so it having the rich in the vitamin C and the flavonoid. So it also shown the improvement of neurological functions. So here the methodology. So we are collected the collection of CPP. The citrus pulp powder was collected from Mew ingredients, one of the industry in Tamil Nadu. So the powder was collected and the powder was subjected to phytochemical screening, and we are done the phytochemical screening by referring the Kokate book, and the acute oral toxicity study was done using the OECD guidelines 203, and it is adopted in 2019 June. And the novel tank test, the behavioral study was done using the novel tank test. So we are group. So here normal, control, standard, and the 3 different doses of citrus pulp powder, that is 50 mg, 100 mg, and 200 mg. So it is selected depend upon the acute toxicity study. So this is a schematic diagram of CPP treatment. Here, the every 3 days we have exchanging our water, and at the 7th day we are conducted the NTT test, and at the 4th day we are conducted the acute oral toxicity up to 4 days. So day 1, day 2, day 3, and day 4. So here we are calculated the mortality percentage, and at the 9th day we are conducted the biochemical parameters and the animal sacrificed and grain was collected, and we are evaluated the stable and MDA. From this methodology, we got the result. First one is the phytochemical investigation. So here we found that the tannin, alkaloid, and flavonoids having a rich in our sample, and the acute oral toxicity was done for 4 days. So here we are collected the percentage mortality at first day, second day, third day, and fourth day, and we found that 312 has shown the lethal dose parameters was shown in the our citrus pulp powder. That is the number of entry on the top and number of time spent in the top is significantly increases as increase in the concentration. So and we also found that the CPP 200 mg has shown the more significance, and as well as standard also shown the more significance in the number of entry on the top and time spent in the top, and as well as parameters such as acetylcholine and MDA. So we are here we found that that shows the decreases in the acetylcholine level and MDA level. It increases the CPP. It increases the concentration. So here we found that the standard and 200 mg of our sample has shown the more significance as compared to normal. So in this study we also done all this comparison with the control normal only. And conclusion and summary. From this we studied the acute toxicity, found that 312 mg per ml is safe to use for the zebrafish, and we also assessed the and evaluated the different parameters, and we shown that the significant increases in the top in the number of entry and number of time spent. And these are the reference which I used for this study. Thank you.
Hello.
Sir.
Banappa sir, zebrafish is your
Yes, sir.
awesome.
I was just happy to see that Sandeep has completed that work. Sandeep?
Yes, sir. Yes, sir.
Were you successful in that conducting that experiment?
Yes, sir.
Where from you got those animals?
Zebrafish, sir.
Yeah.
I'm collected from the Mumbai, sir.
Okay, where from Mumbai?
I don't know, sir. One of my friend brought this.
Okay, okay. So, have you designed that the tank, or how you have measured those parameters?
I'm designing the novel tank, sir, and YMH. But here I didn't mention the YMH. Only I mentioned here novel tank. This is why me only designed by this. From it means buyers from this wood made shop. From that I designed, sir.
So for the fish studies, no, how can you use those wood made?
Not sir, not wood made. This is glass made, but from the that wood made shop, I collected the glass made for the cutting and all those things. So.
Okay. It's okay. So I'm very much happy that you have completed that study. It's okay. I think that study is done in a very good manner. It's a novel attempt. So Chandrasekhar sir, you can ask if any. Guys, over to the moderators for the next presentation.
Next up, we have Namita with track ID number T1PCOL 016. I repeat, T1PCOL 016.
Under the guidance of our principal lecturer of pharmacology department that will be continued. So, what is osteoporosis? Osteoporosis can be defined as systemic clinical disease characterized by low bone and microarchitectural deterioration of bone tissue, the consequent increase in fragility and capacity. In early ages, osteoporosis is not usually diagnosed. It is not. It does not become clinically effective. Even though most of the advanced stage and basic fracture include multiple stage, here in that the economic side of potential is huge. This popular practice is tropical and subtropical areas with nutritional benefits evidence of increased nutrition. Sweet potatoes are rich in carbohydrates, dietary fibers, beta carotene, also vitamin B. It was indicated that calcium is responsible for vitamin. The plan of my study tends to contain rich concentration of. However, there are no scientific reports in the literature of this plant having anti-osteoporotic activity. Keeping all this view in mind, the methodology. Preparation of the ethanol instead of ethanol method was carried out by using the extraction, that is, chocolate extraction using ethanol, 70% volume by volume. Phytochemical analysis, that is, preliminary phytochemical analysis carried out. Experimental analysis, that is, 30 milliliter bag of 3 months old, weighing 1 to 20 grams of the liquid and divided into 5 with comprising of standard. Osteoporosis is administered by using nitroglycerin at 7 kg body weight up to units, once in a week up to 30 days, and 1000 IU per kg of alpha-cholic apparently once a day, daily for 25 days. And the treatment period was followed by the administration of ethylenic potatoes, lamb liver, bird or virus for 30 days of the experience. Evaluation of anti-osteoporotic activity, physical and biochemical estimation, like physical estimation like tumor weight, volume, and density was carried out using the accurate principle. Serum and urine analysis was carried out using ERPH. And this here are the few results of the effect of ethanolic extract of lithium butter. This graph shows the result of FEMA weight, which was in the group of animals infused with supercorticoid osteoporosis treatment against ethanolic extract of lithium butter. This is the effect of ethanolic extract of mare potatoes over serum alkaline phosphatase. This is serum calcium and phosphorus. Here are the graphs of heparin induced osteoporosis. The main discussion or result we come to is the ethanolic extract of mare potatoes lamb liver has exhibited several phytochemical constants such as carbohydrates, levanin, tannins, and caffeine. Treatment with ethanolically mare lamb liver at a dose of 400 mg per kg altered bone weight, volume, also dense, and also showed significant changes in biochemical parameters like alkaline phosphatase, serum calcium, serum phosphorus, urine calcium, urine phosphorus. Also, the people extra additionally possess antioxidant activity with IC50 values, that is, NDP challenge activity. Conclusion is ethanolic extract of mare lamb liver with good antioxidant activity along with antioxidant activity. It can be used as a combination therapy with potent novel antioxidant molecules. Here are few references for this theory. Thank you.
Hello.
Namita, please unmute yourself and turn on your video. Now we can proceed with the Q&A session.
Namita, it's a good work and good presentation. So can you elaborate that what is the rationale behind selecting this plan?
So the sweet potato, that is Ichmea batatas lamb tubers, that is we are used from the traditional period only. So that contains rich nutrients, and also it contains rich in calcium, which is majorly responsible for less calcium is which leads to osteoporosis. So sweet potato contains high amount of calcium. That's why I chose it. Sir.
Okay. So the results are having the translational value, which is readily available and consumable nutraceutical.
Yes, sir.
Okay. Congratulations. Good work.
Thank you, sir. Thank you so much.
Over to the moderators.
Yes, okay. We will proceed to the next presentation. That is Dr. Karthike with track ID number T1PCOL 015. T1 PCOL 015.
Myself, Dr. Karthikeyan, working as an associate professor at Nirmala College of Pharmacy, Mattura, Karnataka. Today I'm going to present my preliminary research work. My main motto behind this work is Krupacon. I want to develop a Krupacon sensitivity and its molecular level activity. So here, before going for that, I would like to work with that of the preliminary work with Krupacon. As all we know that it's a well-known compound actually. So it's a Krupacon. So my title of work is In vitro cytotoxicity activity and cell cycle analysis of Krupacon in lung cancer cells. So just before going for my description of my work, I would like to give a brief introduction. That is, a Krupacon is a well-known chrysopenic acid. It is also known as chrysopenic acid, and it is a naturally occurring anthracene, and it is found in various medicinal plants. Here, it is also been widely used for food and pharmaceutical industry. So, before if you see that the pharmacological effect of the Krupacon, it is having, including the neuroprotection, antibacterial, antiviral, antioxidant, and it's also having used in the treatment of asthma and diabetic complications, as well as Alzheimer's, and also various types of inflammatory response. So the main aim of this work is to study the in vitro cytotoxicity activity and cell cycle analysis of Krupacon in lung cancer cells. It is as a preliminary work. And the methods what we follow here is this: we are selected that of a cell line is a lung cancer cell line, that is A549, and cytotoxicity of the Krupacon is carried out in this dye reduction assay, that is a well-known one, that is NPT assay, and the death of the same has been cell death has been determined by the field what is the stages of apoptosis by the annexin 5. So here, by that of your flow cytometry, the cell cycle analysis also carried out and by with the help of the flow cytometry. And just I will be shift to my work towards what is the result I got after this methods what I followed here. We can see that of the diagram which shows that of the control and the etoposide and the Krupacon. So by saying that we are having a good idea what's the viability of the cells which is present after this entity assay. So we see that in case of cell alone, that is a control. You see it is 100% of cells are viable here, and in case of the etoposide, if we say at 100 microgram 26.70, and etoposide at 150 microgram is 24.35, and 200 microgram is about 13.56%. As we know that if we see that the concentration increases, the viability of the cells is decreasing. It is shown as like here, if you see the Krupacon, and we have started for the both the Krupacon as etoposide as the same concentration at 25 microgram to 200 microgram. But in case of a space adjustment, just we are shown that the important field, a higher concentration is shown here. That is 100 microgram. You see, in case of an Krupacon, it is 52.31%. The cells are viable. And in case of 150 microgram, 50.31%. The cells are viable. And 200 microgram, 39.09%. As like compared to the topic of suicide, this also shows that when the concentration increases, the cell viability is getting decreased. In case of at a 200%, in case of etoposide, we see that it is showing about 13.56% of cells are viable, but in case of the Krupacon, 39.09% cells are viable. So it shows that anti-cancer activity or cytotoxic, the basic acid reacts with the kind of anti-cancer. And here we see that of apoptosis studies by the flow cytometry, we see that in case first graph is shown is it is given as a program, it is shown as a Krupacon, and in case of etoposide as well as a topic. So we see that the Krupacon, it is showing that of your late phase apoptosis, a higher percentage cells at a late phase apoptosis, but in case of the apoptosis says in case of the etoposide, it is in different stage. It is most of the cells are shown in this stage. And here, in case of control, if you see that the cells, such as most of the cells, 99% of the cells are viable at the light phase. So these are the things. By this, we are able to see that apoptosis and the etoposide, that is apoptosis, are different in case of Krupacon as well as in the case of etoposide. So finally, we see that cell cycle analysis, what are the different phases that has been get arrested? You can see, so in case of the, by this we are able to conclude by seeing that of the control as well as etoposide as a Krupacon, if you see that, so the Krupacon is arrested at Z0, Z1 phase as like the etoposide. So by this, I would like to conclude my finding that is Krupacon having a significant anti-cancer activity by doing all these three methods. And further research of molecular level pathway has to be done, and also followed by in vivo as studies has to be done to carry out the finding of the anti-cancer activity. So our aim is to develop and Krupacon derivative, synthetic derivative. So it gives a basic idea of what is the activity of the Krupacon, and it shows a very good anti-cancer activity. So we are going to further and expand our studies for the synthetic derivatives. So by this, I would like to conclude, and I would like to thank the Kripnadevi College of Pharmacy to giving a chance to present this work. Thank you. Thank you so much.
Good presentation, Dr. Karthikeyan.
Yes, sir. Yes, sir. Yeah, myself.
So, what is the future plans of this work?
Actually, past 10 years I'm working with the plant only, but this year I made an attempt to just go for the semi-synthetic derivatives, sir, with Krupacon, and some other compounds also we are selected of our own interest in our lab. And this is a preliminary work we are conducted here. And because already the research was gone with the same Krupacon, with the same cell lines with different targets like necrosis and everything. But in our focus is towards sort of your apoptosis in different pathways in the caspase and others. So this is a preliminary work in our lab we are done, and so we meet success and found to be activity. And we know that that Krupacon is already having proved to have so many activities. But this is one of the compound which is highly available in most of the plants, at least in the basically if you see within aloe vera also it is available.
Yes, it's
yeah. Yes, sir.
Yeah, it's a readily abundant chemical in most of these.
Yes, sir. Yes, sir. Yeah.
It's a good activity. Congratulations.
Thank you, sir. Thank you, sir.
To the moderator if there are no questions.
Chandrasekhar sir, do you have any questions?
Madam, no questions, please.
Okay.
Okay, thank you. Thank you.
Next up, we have Manjima with the track ID number T1PCOL 017. I repeat, T1PCOL 017.
Hello everyone, my name is Vandana Devan. I'm from Karnataka College of Pharmacy, and today I'm going to present a poster on the topic anticipated effect of communication in health. So as you know, psychosis is a mental disorder which can occur due to various reasons like some psychiatric condition such as schizophrenia or other mental disorders, or due to some medication or drug. So it occurs in the late adolescence or early adulthood, and it continues in the adult life. So uh, uh, already various psychotic drugs have already developed, but it has so many side effects like dizziness, then diabetes, weight gain, etc. So to overcome this kind of problems, it is very necessary to develop the organic source of treatment using natural herbs or supplement. So here I am using Benincasa hispida, which is belong to the Cucurbitaceae family, and the major constituents of Benincasa hispida are volatile oil flavonoids and glycosides, saccharides, etc. And the pharmacological study has already revealed that it has so many pharmacological activities which include central nervous effect, then antioxidant, antihistaminic, analgesic, diuretics, neuroprotective, etc. Now coming to the objective of my study, so the objective is to estimate the antioxidant enzyme level in the brain tissue and to explore the antipsychotic activity of Benincasa hispida group. And coming to the methodologies, so here I am using Mr. Red weighing 200 to 250 gram, and then psychosis is induced in the animals by L-DOPA at a dose of 25 mg per kg of body weight orally. And then coming to the experimental model, so here I have used in vivo model, that is two in vivo model I have used. One is epilepsy in mutant and second one is phase suspension based, and the biochemical analysis has been done. That is estimation of antioxidant enzyme level in the brain tissues. So here I have done estimation of catalyst, estimation of reduced glutathione, estimation of superoxide dismutase, and estimation of lipid peroxidase. So I have divided the animals into 5 groups, and there 6 animals each group. So group 1 is the normal control group, where I have given only saline at a dose of 10 ml per kg of body weight orally to the animals for 14 days, and group 2 is the disease control group, where I have given L-DOPA at a dose of 25 mg per kg of body weight orally for 7 days, and the group 3 is the standard group, where I have given haloperidol at a dose of 4 mg per kg body weight intraperitoneally for 14 days, along with L-DOPA at a dose of 25 mg per kg body weight orally for 7 days. Then the group the fourth is a test group where low dose of Benincasa hispida has been given, that is 200 mg per kg of body weight orally, along with L-DOPA at a dose of 25 mg per kg of body weight orally for 7 days. And group 5 is also test group where high dose of Benincasa hispida, that is 200 mg per kg of body weight is given orally, along with L-DOPA at a dose of 20 mg per kg of body weight. Now coming to the result part, so data was analyzed by using one ANOVA, and the p-value is determined by comparing the test group with a disease control group and the disease control group with the normal control group. So from this, we can say that that is drug that is given as a risky that there is antipsychotic activity in a dose-dependent manner against psychosis, which is induced by L-DOPA. So, from the result, you can see that there was a significant decrease in duration of immobility and increase in duration of catalepsy in the rats, which determined that this drug possesses antipsychotic activity. And we can also say that there was an inhibition of lipid peroxidation and rise in the glutathione, then superoxide dismutase and catalase level, which indicates that this drug also possesses antioxidant property. So, from this study, we can demonstrate that the thyroid extract from the Vesica possesses antipsychotic activity, but further studies are required to identify the active principle which is responsible.
Now we can proceed to the question and answer round.
Dr. Chandrasekhar sir, you can have questions. I could not hear and see properly anything. So.
Yeah, same thing. Hello.
Hello, good afternoon, sir.
Good afternoon. There are because the clarity was not there in the voice and slides. Okay, but even though, tell me what is the basis of selection of this kind of work?
Sir, because antipsychotic drugs, many antipsychotic drugs are already developed, but I have chosen this natural herb, which have less side effect compared to the antipsychotic drugs, and the fruit have many phytoconstituents like flavonoid, 10-quercetin, and vitamin C, B. So they possess central nervous effect and antioxidant property. So based on this, I have chosen this drug.
This plant is very well known for availability, and it is consumed everywhere.
Yes, sir.
Okay, good luck. Thank you.
Thank you, sir.
Next up, we have Drishti Rana with track ID number T1PCOL 0121. T1PCOL 021.
Tirana from Madhuba Pharmacy College, Bangalore. Here I represent the role of clinical pharmacists to optimize the therapy for cancer. Cancer is a group of diseases involving abnormal cell growth with the potential to invade and spread to other part of the body. It is life-threatening worldwide. Now, pharmacists play an important role in the complexity of prescribing and transcribing drug regimens. A pharmacist's role is not only in safe handling, preparation, and dispensing of drugs, but also in pharmaceutical care that improve a patient's quality of life. This process helps to support optimal anti-cancer drug treatment. Now, how to manage medication? Now, most of the anti-cancer agents are targeted therapies, or in the other words, drug whose development is based on the predetermined tumorous or endogenous factors. These drugs are either monoclonal antibodies that interact with cell membrane receptor or circulating ligand or protein enzyme inhibitor that interfere with various tumoral signaling pathways. Oncology pharmacists provide appropriate dosage, administration times, sequences to utilize chemoprotective agents, high dose amifotericin and dexazotene. Now let's consider a patient who is undergoing chemotherapy with hypertension. After full diagnosis of patient, optimize treatment of hypertension. Moreover, we have to give prescription of appropriate chemotherapy and regular pressure monitoring during chemotherapy up to 140/90 mmHg. If complication arise like blood pressure is more than 140/90 mmHg, then provide with AC inhibitor and calcium channel blocker. So, in hospital, in absence of doctor, oncology pharmacists play an important role in drug selection, providing proper medication and good combination therapy. Now, I conclude this. In conclusion of review topic is oncology pharmacists contribute a major role in safety of anti-neoplastic medication. Order device that its therapeutic potential. With the understanding and recognition of drug interaction and side effect, pharmacists can provide timely intervention and information to health providers as well as counseling to patients. So every hospital should be a clinical pharmacist optimizing the cancer therapy for better treatment of patients. And we stop the worsening of patients. Thank you.
We can proceed with the Q&A session.
Good, it's one of the review. There is no much question on this. Can you go ahead with the next presentation, madam?
Sure, sir. Next up, we have Harshita. This is also a review paper with the track ID number T1PCOL 022. I repeat, T1PCOL 022.
Harshita Ashit Shah. Hello everyone, myself Harshita Ashit Shah, Department of Pharmacology and Pharmacy Practice, Manipur Pharmacy College, Uga Tertiary University, Bardoli, Surat, Gujarat. My today's topic is the emerging role of artificial intelligence in cancer drug development and precision therapy. What is artificial intelligence? Artificial intelligence refers to intelligence shown by machine made by humans. Deep learning can find information in image that the human eye cannot recognize. This is great significance for early diagnosis of tumor based on image data. Intelligence can also help diagnosis and treat tumor. Artificial intelligence is often based on multi-layer neural network structure with strong logical reasoning ability and learning ability, which can highly stimulate human thinking mode. Main purpose of artificial intelligence is integration of artificial intelligence technique in cancer care could improve the accuracy and speed of diagnosis, aid clinical decision, and lead to better health outcome. Artificial intelligence guides clinical care and potential to play an important role in reducing health disparities, particularly in low resource setting. This have some results. It helped doctors to correct treatment decision, reduce unnecessary surgeries. Importance of artificial intelligence in anti-cancer development. Artificial intelligence can be used to predict anti-cancer drug activity or assist in anti-cancer drug development. Different cancers and the same drug have different reaction modes. Machine learned models were proved to successfully predict the drug sensitivity of patients. artificial intelligence in chemotherapy session. In this field of cancer chemotherapy, artificial intelligence focused more on response between patients and drugs. The main applications achievement of artificial intelligence include management of chemotherapy drug used. Artificial intelligence can perfect and accurate the optimization process of combined chemotherapy. Artificial intelligence reduced over-treatment of cancer, includes an algorithm that can analyze the digital image of woman's cervix, a machine learning tool that can reduce the excess treatment of lesions suspected of breast cancer. This graph shows the more artificial intelligence is used in neuroplasm than cardiovascular and other diseases. Most cancers are always lung cancers, including breast cancer, liver cancer, etc. By this, I conclude that artificial intelligence has made a unique contribution in anti-cancer drug development and treatment. Humans are limited by their own level of knowledge. It can provide important insights and information that cannot be found by human identification and personalized treatment of each cancer. We believe that artificial intelligence will bring profound challenge to medical technology in future. Thank you.
We can proceed up with the Q&A session if there are any. If there are no questions, we can wind up with the session, sir. Sir? Now we conclude the e-poster presentation of Track 1, Recent Advanced Pharmacological and Clinical Studies. I would like to express my sincere gratitude to the evaluators, Dr. Banappa and Dr. Chandrasekhar, for taking out your valuable time for evaluating the posters. Thank you, sir. And I would also like to extend my thanks to the presenters. Results will be announced in a while during the validatory program. Thank you.
Thank you. Thank you, one and all.
The validatory program will be conducted in the auditorium. Please join through the webinar link. Thank you.